Live streaming is on. Okay, so this was our DNA project. Okay, so it is the part 2 of the DNA project, and previously we have created a dna.py, that dna.py class, and this we have created a DNA class. Okay, and in this class we have this following property: sequence, number of adenosine, number of thymine, number of cytosine, number of guanines. Okay, so these are basically the four bases these are basically the four bases from which DNA is made up of. And this is the length. So every DNA has some length. So this is the length property of the DNA. And again we have one more property, that is each valid sequence. And so this each valid sequence is basically linked to the, if suppose that if dictionary should have only this allowed base. Okay, so A, T or G and C should be present in the dictionary. If suppose that in some scenario the dictionary present in the bases present in the DNA are not AT or GC, then this type of DNA will be called as invalid DNA sequence. Okay, so we have a property to check that is valid sequence. And now again we have complementary sequence. So complementary sequence is nothing but every DNA has a complementary base. Okay, so basically DNA is made up of two DNA is a structure that is a double helix structure, and one helix is basically the complementary of another. So this is how a DNA is made up of. Suppose we have A will be paired with T. Similarly, T will be paired with A. Means adenosine will be paired with T, thymine will be paired with A, guanine will be paired with C, and cytosine will be paired with G. So this this make a concept of complementary sequence. So if we have this DNA, then the complementary sequence of this will be this. Okay. So now we have written. Okay, so we have written this class, DNA class. Okay, now we need to this is a simple DNA class, and here we have functions to get length and to check is valid, and get number of thymine, get sequence, get complementary sequence. So now I basically I given an assignment to this is basically the method that is used for creating the complementary sequence of the DNA. So I given you task to make this method more efficient. So some of the members have practiced on it. Okay, so I'm skipping this. So one solution is already present, that is based on dictionary, and that is based on dictionary and comprehension. So we can use that. And basically, if we tell that in terms of memory consumption, that program is not efficient. Okay, so we will not. I'm not giving you. The solution of it right now. Before that, I am giving you what task we need to perform in this project. Okay, so please hold for 1 minute. Hello. Now we will first we will discuss what task we need to perform on this project. First is we have already taken get complementary sequence. Now I am adding a sample FASTA file. Okay. Always DNA sequences are stored in a FASTA file. So I am creating a sample sequence dot FASTA. Okay. And we will have here DNA sequence one. Okay. Suppose that we have suppose that we have 10 DNA sequence here. Okay, so this is DNA sequence 1, this is DNA sequence 2, sequence 3, DNA sequence 4, DNA sequence 5, DNA sequence 6. Okay, so I am doing some changes here. Suppose that okay, we have this 6 DNA sequence. Okay, so every DNA sequence, first this is the name of the DNA sequence, and this is the sequence. Okay, so first, our task is to create a dictionary to store all this dictionary from a file. Okay, from a FASTA file. So, our first task is to store all the all the DNA present in the sample sequence dot FASTA file into a dictionary. Okay, so our first task is to store all the DNA sequence in the sample sequence or FASTA file in a dictionary. Okay, okay. Keys will be this sequence name, and value will be sequence. So keys will be sequence name and value will be sequence. Okay, so this is our first task. Second is to convert all the sequences in a single line. Basically why we are doing it, so suppose that if we have this DNA sequence, okay, so we want that this DNA sequence should be in a single line. Okay, because there are no logical break between two DNA in logical break in DNA sequence. Okay, so we will store our value like it. Okay, to convert all the sequence in a single line and store in result dot FASTA file. Okay, so I'm giving you example result file. So suppose that we have a result dot dot FASTA file. Okay, so first sequence should be stored in this file like this. Means we will remove the new line from this dictionary, from this DNA sequence. Did you get me, everybody?
Yes, sir.
Okay, so this is our second task. Now Third task is to create a new complementary FASTA file to store all the complementary sequence of of the of the DNAs present in sample sequence.fasta file. Okay, this is our first task to store all the DNA present in the sample sequence.fasta file in a first we will store in a dictionary, and then we will convert all the sequence in a single line. Third is to create a new complementary FASTA file to store all the complementary sequence of DNA present in the sample sequence.fasta file. Okay, is this clear, everyone?
Yes, sir.
Okay, so this is our third task, and our fourth task is really complex. Okay, so our fourth task is based on codon. Okay, so what are codons? So codons are basically, suppose that we will so basically you know that in chromosome basically DNA are present. Okay, DNAs are present in chromosome, and DNAs is transcribed into RNA. Okay, so this transcription is performed on the basis of codon. Okay, so what is codon? So codon are basically combination of three base pairs. Okay, so this will be splitted into, first you need to split this in a 3-3 pair. Okay, so suppose that we are storing, first we have ATG, then we have CTA, and then we will have TTT. Okay, and then we will have TAT. Okay, it is clear, everyone? So the codons present in this DNA will be ATG, and then means here ATG, and then we have means split into 3-3 pairs pair CTA, and then we have TTT. Okay, and we have again TAT. So these are the codons. Okay, so how much codons, how many type of codon present can be present in this in a DNA? Can anybody answer? So this is a simple permutation combination question. Okay, so with how many times, there are basically four base pairs. Four base pairs can be present in a DNA: A, T, G, and C. So on the basis of these four base pairs, we need to create combination of three. So in the first place we have four options, again in the second place we have four options, again in the third place we have four options. So we can create 64 codons. Okay, so 64 codons are possible. So if we create a codon array, okay, so suppose that these are the codons: A, then A, B, then AAC. So these are basically the codons. Okay, so like this we can create 64 codons. So first your task is to create to create and display all the possible codons. Fifth, okay. So create a list. So what will be better to? Okay, so you need to create all the possible codons, and the fifth task is to get all the codons present in the disk DNA. Is clear, everyone? Okay, so Shreya, your fourth point is actually fourth is to create and display all the possible codons. Okay, all the possible codons in DNA. So all the possible codons will be. I told you that there are 64 possible codons. Okay, so you need to identify all the possible codons and you need to print that. Okay, so there will be 64 codons and you need to display all the possible codons, and your fourth task is your fifth task is to get all the codons present in the DNA. So suppose that if we take the example of if you take the example of this DNA, so how many codons are present here? First is ATG, and this is CTA. Third is TTT, and fourth one is TAT. Similarly, fifth one is ATA. So these are the codons that are present in the DNA. So basically, what basically in cell we have first DNA, then DNA is transcribed into mRNA. Okay, and then that mRNA is basically called as messenger mRNA, messenger RNA. Okay, and that messenger RNA is translated into proteins. Okay, and that protein is basically performing all the tasks that are happening in your body. So this is how the flow work in our body. So these information basically the information present in your chromosome, that is in form of DNA, and that DNA is split into three pairs. Okay, so these three pairs are called as codons, and these codons are transcribed into an amino acid. So this all so every codon is basically transcribed into a different amino acid. So you don't need to go there in deep. So you just need to identify these codons present in the DNA. Now it is clear, everyone? Shreya, fourth point is clear or not?
Yes, sir.
Okay. So these are the tasks you need to perform. Okay. Yeah. One more task is that one more task is called as crossing. Okay. So you can call this as crossover. Okay, so what is crossover basically? uh, First I want to show you. Yeah, you can see that this is basically called as crossover. Okay, so suppose that if you have any DNA sequence. Okay, so this is our DNA sequence, this one is our first DNA, and this is our second DNA. Okay, Then crossover basically creates the DNA like this. Means some part of it will be present in this. You can see that this part of DNA is split into this. Means in this DNA now we will have this part and half. Suppose that you are splitting in 3 is to 4 and 1 is to 2. Okay, so suppose that now we are splitting half, so half of the sequence will be present from its previous and half will be from the another. So crossover always performs between two DNA. So basically the crossover is performed in two DNAs, and this DNA will result into a different DNA. This is how the information is transmitted into a species. Okay, so suppose that if we have two different species and they are combining, so then they cross over, their DNA crossover, and they result into a different type of DNA. Okay, and that result into a different type of property, different type of protein, and that protein results into a different type of human, different type of species. So this is so suppose that if all of us has same DNA, then our all the property will all the man will be same. But what happens if man and means basically when the crossover occurs, that that basically cause the difference between two persons and two species. Okay, so we need to perform the crossover. So whenever we perform the crossover, we will have two DNA. Okay, and we will suppose that we are performing the crossover. So in the programming context, how will it present? Suppose that I am taking two DNA sequence from this file. So first I am taking DNA sequence 1 and DNA sequence 2. Okay. So we have first this DNA sequence, and suppose that we have again a different DNA sequence that is this. Okay, now we are performing the crossover. Okay, so, so whenever you perform a crossover, I will give you that crossover will happen. Crossover will happen with 2, 3 or 5, 6. So it means that first DNA sequence will combine into, so crossover, if I am telling you that crossover will happen at 5, 6, it means that suppose that I am removing this, okay, this from here. So it means that 5, 6 means that 1, 2, 3, 4, 5. So fifth, and this is sixth. Okay, so crossover will happen. Crossover will start from here. Okay, so 5, 6 means fifth from the fifth index. This is the fifth index of the DNA, and this is the sixth index of this DNA. So crossover is starting from this. So our resulted DNA will be this. It's clear, everyone? Okay, crossover part is not clear, and others I will discuss with you. Okay, so if you are facing the issue, I will resolve it. Okay, before closing the today's class, I will uh, I will I will provide you a solution. Okay, so yeah, crossover is not clear. So I told you that first we will have two DNA sequence. Okay, this is our This is our first DNA sequence, and this is our second DNA sequence. So, and our I am told you that crossover will happen at 5 6. So first you will go to the fifth index. So what is the fifth index? 8. This is 0 1 2 3 4 5. So this is 5, and the sixth index for second, 1 2 3 4 5 6. So this is the sixth index of, sorry, this is the fifth index, and this is the sixth index of this. Okay, so I've told you that crossover will happen at 5 6. It means that first you need to take this part from DNA sequence 1 and this part from DNA sequence 2. Okay. And for second crossover part, you will take this part from this, from second, and this part from first. And after you need to combine this. Is this clear, everyone? Yeah. So this is how crossover will happen. So you need to also perform the crossover. Okay. So to perform the crossover, you will, so suppose that we have 5 DNA sequence. So you need to perform the crossover between all of them. So okay, and crossover will happen in every case. In every case, the crossover will happen at 5 6. Okay, so your four task is, so your four task is to perform crossover, to perform crossover between every every pair of DNA sequence present in, between every pair of DNA sequence present in sample sequence.fasta file. So this was our sixth task. So pairing is nothing but selecting. So I'm telling that to perform crossover between every pair of DNA, every possible pair. So I'm saying you that every possible pair. Okay. So every possible pair means what? So we have DNA sequence 1, DNA sequence 2. These can combine. This can have yet we can have crossover. So again, similarly, you will you will never perform crossover between same sequence. There is no meaning of crossover between same sequence. Okay, between two same DNA sequence. Okay, so you need to suppose that here we have five first second, so we have six DNA, then six C2, that is basically 6 star 5 divided by 2. So this much of pair can be done. So suppose that we have 15 pairs. So 15 pairs can be possible, and you need to perform the crossover between these 15 pairs. And you need to, so suppose if you face this as difficult, then you just create a sample sequence 2 dot faster, okay, and perform crossover between them. Is this clear now? Yeah, yeah, permutation combination. So hello. So do you want me to write the code, or you can practice by yourself? Nobody is answering. Yeah, I also want that you try by yourself first. Okay, so if you can't write the code, then I will write the code in next class. So these are the basic tasks, and one more task I want to assign. Okay, so that is basically called as mutation. So one more thing that occurs in DNA is called as mutation. Okay, so suppose that. So what is mutation? First, I want to show you mutation. So suppose that if you are exposing to DNA, exposing to UV, okay, ultraviolet rays, okay, so that damages your DNA. So in that case, what happens basically, some part of your DNA is modified, okay, and that is called as mutation, and your base pair will be, then DNA present in your base pair will be not only AT and GC, there will be some extra base pairs that will be unwanted DNA, and that is basically responsible for causing different behaviors. Basically mutations are good and bad also. So if mutation happens some functional part of your body, then that will cause you some serious problem in your body. Uh, but this rarely happen, and when this mutation is occurred in your body, then basically it is converted into some serious problems, and one part of serious problem is cancer happens due to mutation. Okay, so you don't need to go there. So basically we have mutation that happens in DNA, so we will also perform mutation. So you can see that mutation always occurs in some part of DNA, so I will give you a position to perform mutation on that part. Like corona mutation. Yeah, this is also called as, yeah, this corona mutation is also linked to the mutation. So in some of the coronavirus mutation has occurred. So that mutation is good for that coronavirus, but it is not good for us. Okay, so this is mutation. So some part of DNA has been modified, but we will also perform that mutation in our project. Is this clear, everyone? Okay, so suppose that, so I'm keeping this mutation very simple. Suppose that we have this DNA sequence. So suppose that if we have this DNA sequence and we want to perform the mutation, so I will give you the, so you need to create method for it, okay, to perform mutation, and I will give you the position, or index at which mutation need to be done. Okay, and whenever you perform mutation, so you need to replace the base pair by M. Okay, so suppose that if I am telling you perform mutation at index 2. Okay, index 2, and replace every mutation by M. Okay, so what you will do, two index is this, you will replace, so this DNA will be converted into DNA will be converted into this. No, 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 this. Okay, so we will have here C also. Yeah. So this DNA will be converted into two. So suppose that if I am giving you the index 2, 3, 4. So all the base present in 2, 3, 4 should be mutated to M. Okay. So, so now this DNA will be converted into all the bases at 2, 3, 4. So this is 2, this is 3 and this is 4 will be converted into this DNA. So you need to create a method to perform mutation in the DNA. Okay, and your task is to perform mutation in every DNA present in this sample sequence dot FASTA file at position perform mutation in all the sequence present in FASTA file FASTA sample sequence dot FASTA. Okay, FASTA file at position 34 and replaced by M. Is this clear, everyone? Okay, so this much task we will perform in our DNA project and it will be completed. So, okay, so I am not doing anything here. Just I told you that perform mutation in all the sequence present in sample sequence dot FASTA file at position 3 4 and replace the base by M character. Okay, yeah. So So I would like you all submit your answer in next 1 to 2 days. Okay. So we will connect. So today is Thursday. Okay. So we will collect. We will connect on Monday. And if you not perform, if you not give me the solution, then I will give you the solution on Monday. Okay. So I think you will take 2 to 3 4 days to complete this task. So try to use all the class object principles. Okay. Okay, everyone. Is this project cool or should I change anything in this project? It's cool and hard to, yeah, actually this project is linked to bioinformatics and lots of researcher in bioinformatics don't know this. So if you are knowing this, this is very advantageous. And if you know any bioinformatician or any of your friends is reading in if any of your friend is reading in biotechnology, then you can question in this. You can say that I am doing performing coding related to DNA and bioinformatics. Okay, so okay everyone. So one I think Jatin or who was facing issue. I think it was others. Yeah, others was facing issue. So others you can remain, but sir, in which class? Wait. Yeah, DNA is basically started from I think class 9th or 10th. So you will read DNA in class 9th and 10th, and not in that much deep. This mutation crossover will be in the syllabus of 12th. So no, no, no. Actually, this is yeah one more interesting thing in bioinformatics that you don't need to know molecular biology. So you just need to have some basic concept of DNAs and how DNA works. What are the basic properties of DNA? So these are the basic requirements for becoming a bioinformatics. And Deoxyribonucleic acid. Yeah, DNA full form is deoxyribonucleic acid. Okay, so I also have lots of interest in bioinformatics, I'm also the expert of bioinformatics field also. So that's why I am explaining this you all. Okay, so start practicing on it, and we will connect again on Monday. And others, you can stay, and we will discuss your problem. Okay, everyone. Thank you.